When a thunderstorm begins, it shakes the sky, turns heads, and demands attention. Just like that, in this Farmer Thunderstorm series, we're going to create an energy so powerful that nothing gonna stop you. Just like a thunderstorm strikes, every session will be like fast, sharp, and unforgettable. And the sound of your success will echo all the farmer background. So my name is Divanshu and welcome to the channel GDC English. In this video, we're gonna start a new series that is Pharma Thunderstorm. So in this video, we're gonna see some MCQs regarding the general pharmacology, which are very important for you for GPAT exams or NIPER exams or whatever exam that you are preparing for, just like pharmacist. Okay. So in this video, we're gonna see some important MCQs. So let's get started. So the first question is, who is the father of pharmacology? Option is Thomas Hobbes, Gregor Mendel, Herodotus, Oswald Schimmelberg. Thomas Hobbes, Gregor Mendel, Herodotus, and Oswald Schimmelberg. So you have to answer in 5 seconds. So the answer is Oswald Schimmelberg. The Oswald Schimmelberg is the father of pharmacology. So can you name me who is the father of Indian pharmacology? Hmm. Tell me in the comment section, okay? I will look onto it. So this is the explanation that what is the father of who is the father of modern pharmacology? That is Oswald Schimmelberg. So in this table only you will find many of the questions. Many of the questions has been asked in various exams, previous exams. One of a kind like who is the father of chemotherapy? The answer was the Paul Ehrlich. Who is the discoverer of penicillin? Alexander Fleming. Who is the discoverer of insulin? Banting and Best. Okay, so this table is very important for you to learn. So next question is characteristic of highly ionized drug. First one is excreted mainly by the kidney, can cross the placenta barrier easily, is well absorbed from the intestine, accumulates in the cellular lipids. So you have 5 seconds to answer. Answer. So the answer is excreted by the kidney. Okay. By because whenever a drug is highly ionized, whenever a drug has a charge, it will automatically have a water solubility capability in that particular compound. Okay, so whenever a compound has ions in it on it, so it has mainly water soluble ability. So if it is water soluble, what will happen? It will mainly excreted by the kidney. Apart from that, whenever a drug has a lipid solubility capability, it will easily cross through placental barrier. It will absorb from the intestine. You have read also before that whenever a drug is lipid soluble, it has to be absorbed from intestine. Whenever a drug is water soluble, it has to be absorbed in to the stomach. Or whenever a drug is lipid soluble, it is accumulates into the fat also. Next question: which of the following is not the prodrug? Mercaptopurine, dipiferine, enalapril, or linezolid? So you have 5 seconds to answer. Okay, so let's see the answer. The answer is linezolid because linezolid is not a prodrug; it is an active drug. Okay, mercaptopurine has a active metabolite. Dipiferine, yes, epinephrine. Okay, enalapril, yes, enalaprelate. Enalaprelate is a active metabolite of enalapril. Binding order of drug to tissue is. So these are the options through which you have to take that which organ has the highest binding drug capability. Okay. Kidney, liver, lungs, muscles, muscles, kidney, lungs, liver, kidney, lungs, muscles, liver, lungs, kidney, muscles. Okay. So answer. So the answer is liver, kidney, lungs, muscle. Okay, so liver has the most highest binding capability towards the drug. Then kidney, then lungs, and then muscles. Okay. So what does potency refers to? Potency of a drug refers to what? Okay, affinity of drug to binds to receptor, Affinity of drug binds to receptor and activates it. Dose that requires to produce response. Maximum response a drug can produce. Okay. So what would be the answer? Answer is dose that requires to produce response. Okay. So it is the minimum dose. Potent drug. What is a potent drug? Potent drug is just like a drug which can be used in a very small amount and still it has a good response, good biological, pharmacological response. Okay. So for example, you have to take two drugs, drug A and drug B. Okay, so drug A is clinically available at 10 mg concentration, whereas second drug B, is available at 50 mg at concentration. Okay, so you tell me which drug is more potent? So your answer will be drug A because the drug A is usually consumed at less concentration as compared to the other drug, which is 50 mg, and whereas 10 mg drug A is usually being taken. Then that drug A is usually being taken as 10 mg. That is also showing that biological response. Okay, so that drug will be a more potent drug. Okay. All of the following drug induces microsomal enzymes. So where are microsomal enzymes present? Tell me first. Okay. So where microsomal enzymes presents? You have to tell me in the comment section. Except. Okay. So which drug is a microsomal inducer? Okay. So in this question, the question is being asked that which drug is usually have a inducer capability, microsomal enzyme inducer capability, that activates more enzyme, that it activates more drug metabolism of other drug. For example, a drug A is increasing drug metabolism of drug B. Okay, so what it is doing? It is actually increasing the metabolism of drug B. How? Because it increasing the microsomal activity of enzymes. Okay, so which of the drug does not have that ability? That question has asked from you over here. Cimetidine, griseofulvin, rifampicin or phenobarbital? Okay, so the answer is cimetidine. How? Because enzyme inhibitors, these are GPRS cell phone. GPRS cell phone are enzyme inducers. They are increasing the metabolism of other drugs. Okay. Whereas cimetidine is an enzyme inhibitor. It is decreasing the metabolism of other drugs. Okay. And this enzyme inhibitors can cause the toxicity. So in this case, enzyme inducers, the dose of such drug should be increases, whereas in the enzyme inhibitors, the dose of such drugs should be decreases. Let's move on to the other question. Next question, seventh question. First order kinetic means constant amount of drug eliminated per unit time. A constant fraction of the drug in the body is eliminated per unit time. The fraction of administered dose that reaches systemic circulation. The rate of elimination is independent of plasma concentration. Okay. So the answer will be a constant fraction of the drug in the body is eliminated per unit time. So let's have some explanation. In first order kinetics, the rate of drug elimination is directly proportional to the drug concentration into the plasma. This means a fixed percentage, a fraction of drug is removed per unit time, not a fixed amount. If it is asked a fixed amount, that fixed amount will be into the zero order kinetics. Okay, not in the first order kinetics. In the first order kinetics, there is a percentage fraction. Example, if 10% of the drug is eliminated every hour, 10%. Okay, so starting from 10 mg, 10% of 10 mg, 90 mg, 10% of 90 mg, 9 mg. To eliminate 9 mg, 81. 81, 10% of 81 mg, that is 8.1. So minus 81, 81 minus 8.1, it will be around 72.9 and so on. Okay. So that percentage, the fraction, okay. Consider the word, the concentration, the fraction word. Fraction or percentage. Fraction or percentage will be correlated towards first order kinetics. Otherwise, the amount, a particular amount, for example, 20 mg is being eliminated in hour per hour. Okay, a 20 mg drug is eliminated per hour. That will be your zero order kinetics. Understood? Okay. Next question. Type 1 ADR reaction is adverse drug reaction is caused when T cells bind to specific antigen, caused by tissue injury, IgE mediated, caused by cytotoxic antibodies. Answer? Yes, IgE mediated. Yes. So IgE mediated is an anaphylactic reaction, anaphylaxis reaction, which is generally a type 1 reaction. Anaphylactic reaction is also known as the type 1 ADR reaction. It is generally mediated by IgE. Type 4, that is delayed, okay, delayed hypersensitivity reaction, that is caused by T cells bind to a specific antigen. Acha, now you tell me what kind of antibody, Ig antibody, is being uh, used as a cytotoxic antibodies. Okay, if there is a cytotoxic reaction, generally it is a type 3 reaction, type 3 ADR reaction. What kind of antibodies are being used here? You have to give me answer. So next question is, after third half-life, the amount of drug left is, after third half-life, amount of drug left is, okay. For example, you have 100% of drug. Then the third half-life, what amount of drug is left? No, that is a simple addition. Okay. So answer is 12.5. How? First half-life, 50% drug remains. Half-life means 50% left. Then second half-life, 50% of 50% means 50% divided by 2. So it will come around 25%. Then after third half-life, 25 divided by 2. Then it will come around 12.5. So after third half-life, 12.5% of drug is remaining. Okay. So now, action of alpha subunit of G protein is, action of alpha subunit of G protein is, okay, binding of agonist, conversion of GDP to GDP, breakdown of GTP to GDP, internalization of receptors. Okay. So answer must be from these options only. Okay, B or C. Let's see the answer. Answer is breakdown of GTP, guanine triphosphate to guanine diphosphate. Okay. What does 3 in CYP3A4 denote? This question is very important. It has been asked in previous GPAT question, previous GPAT exam. Okay. So what does 3 generally refers? Okay. So options are family, gene number, or allele. Okay. The answer is family. So here you can see the CYP3A4, 3 for family, A for subfamily or gene, 4 for gene number or specific isoform. Okay, so you have to learn this, that 3 signifies family, A signifies gene or subfamily, 4 signifies gene number or specific isoform. Next question, the comparison of bioavailability between two dosage form is referred as. It is very easy question, no? The comparison of bioavailability between two dosage forms. So one is a tablet, one is an IV. So what is the comparison between it? What does term called? Bioavailability, biopharmaceutics, biological, or bioequivalence. Okay. So answer is very simple. That is bioequivalence. Bioavailability is the fraction of drug that reaches into the systemic circulation. It is bioavailability, that is a percent or a fraction of a drug that reaches into the bioavailability. For example, a drug has been taken 100% initially. It's very obvious that whenever you take a drug, it is in 100% form. But whenever you absorb it, whether it is from stomach or it is from intestine, it will go into the first pass metabolism. After that, only 60 to 70% of the drug gets into the systemic circulation. So what was the bioavailability of that drug? Only 60 to 70% is bioavailability. But if we talk about bioequivalence is somewhat like whether that you are comparing a drug into its dosage forms. For example, PCM as a tablet, paracetamol as a tablet, or paracetamol as a IV. So comparison between the tablet and IV is called the bioequivalence. Okay. Bioequivalence studies are also crucial for the generic medicines also. So whenever you um, differentiate a generic medicine towards the branded medicine, bioequivalence studies are also conducted there. Now, next question, which of the following is the correct order in the drugs binding to various plasma proteins? Okay, so there are various kind of plasma protein, albumin, alpha-1 acidic glycoprotein, globulins, or lipoproteins. So what is the correct order? Okay, you have to answer it. Albumin, alpha-1 acid glycoprotein, globulins, lipoproteins, globulins, albumin, globulins, lipoprotein, alpha-1 acid, albumin, alpha-1 acid, lipoprotein, globulin, albumin, lipoprotein, globulin, alpha-1 acid glycoprotein. So it is very much known that albumin is very widely distributed, widely much present inside the body. So albumin should be first. But what about next? That you have to answer. The answer is albumin, then alpha-1 acid glycoprotein, lipoprotein, and then globulins. Now, so drug binds to different plasma protein with different affinity. Albumin stands for basically binds for acidic drug, that is warfarin or aspirin. Alpha-1 acid glycoprotein binds to basic drugs. Globulin, steroid, and metal ions. Lipoproteins, lipophilic drug when albumin binding is saturated. Okay. Acha, wait a minute. So answer must be here. This one, no? This one, not this one. This is not the answer. No, no, no. A is the answer. A is the correct answer. Okay. Not C1. Albumin, alpha-1 acid glycopropene, globulin, and then lipoproteins. Okay. Yes. So answer is A1. Albumin, alpha-1 acid glycoprotein, globulins, and then lipoprotein. This one is the correct order. Albumin, alpha-1 acid glycoprotein, globulin, and then lipoprotein. So lipoprotein has the lowest binding affinity. Next question. True statement regarding inverse agonist. It has been asked in various times. Okay. So binds to the receptor and cause intended action. Binds to the receptor and cause opposite action. Binds to the receptor and cause no action. Binds to the receptor and cause submaximal action. Okay. This is very interesting. So answer it. Which option is right for the term inverse agonist? Yes, for inverse agonist, it will cause an opposite reaction. So what is the intrinsic activity of inverse agonist? You have to tell me in the comment section. What is the intrinsic activity of inverse agonist? Okay, +1, -1, or 0. Okay, so the answer is binds to the receptor and cause opposite action. Okay, so this one binds to the receptor and cause intended action, that is for the agonist. Binds to the receptor and cause opposite action, that is for inverse agonist. Binds to the receptor and cause no action, that is for antagonist. Don't be confused about the intrinsic activity of that drug. Antagonists have intrinsic activity 0 and inverse agonist have inverse agonist have intrinsic activity that is -1, not the antagonist. Okay, binds to the receptor and causes submaximal action, that is between 0 to 1, that is partial agonist. Okay, it has not just like same action like agonist, but it do have some kind of action like agonist, lower than the agonist. So what is the intrinsic activity of partial agonist? That must be around 0 to 1. It may vary around 0 to 1. Agonist binds to receptor activated, produce the expected intended effect. Antagonist binds to receptor, blocks agonist, produces no effect. Some maximal activity is 0. Inverse agonist binds to the same receptor as the agonist but reduces its basal constitutive activity, produces the opposite effect of the agonist. Okay. So phase 2 clinical trial is also called. Yes, it has been asked previously. What is the synonym of phase 2 clinical trial? Therapeutic confirmation comparison, human pharmacology and safety, therapeutic exploration and dose ranging, post-marketing surveillance. Post-marketing surveillance is very much known to you. But what about phase 2 clinical trials? So let's see the answer. That is, phase 2 clinical trials is also known as therapeutic exploration clinical trial. Okay. So what about phase 1? Phase 1 is for human pharmacology and safety. Phase 1 clinical trial is also known as the human pharmacology and safety. Phase third, therapeutic confirmation. Third, 3 and 3 for C, because C is usually come at third position of A, B, C, D, no? So C, third. C, third. Therapeutic confirmation, C, third. Okay. Human pharmacology for first. Therapeutic exploration for second. Post-marketing surveillance, everyone knows, fourth one. Phase 4. Okay. Which of the following is the active metabolite of codeine? Codeine is a opioid. Okay. So options are hyoscine, morphine, atropine, or caffeine. Active metabolite of codeine. So codeine is a opioid, as I've told you before. So among of them, which one is opioid? Morphine, obviously. So the op- correct option is morphine. Maintenance dose of a drug depends primarily on. That is quite good question. Volume of distribution, half-life, lipid solubility, total body clearance, maintenance dose. Okay. Answer. So the answer is total body clearance. Why? Maintenance dose is a dose need to keep a drug concentration as the desired steady state level. So there are two types of dose. Okay. First one is the loading dose, which will be given initially. Okay, which will be given initially just to raise a concentration, just to have a concentration of drug into the blood circulation, but to maintain it. Okay, but to maintain it you have to give a maintenance dose. Okay, so there is a steady dose, steady state concentration. Steady state concentration is a concentration through which the drug will show its effect throughout its duration of action. Okay, so firstly it has to be given a loading dose. After that, there will be a given as maintaining dose, that is maintenance dose. So, what kind of factor this maintenance dose depend on? So the option is clearance. Okay, so whenever there are clearance, that will affect the maintenance dose. Okay, so the clearance determines how fast the drug is removed from the body. So it is the primary factor influencing the maintenance dose rate. Other factors like volume of distribution affect the loading dose, not the maintenance dose. For the loading dose, there is a factor that is responsible, which is volume of distribution. Okay. Maintenance dose depends on clearance. Loading dose depends on volume of distribution. I hope I made you clear. Type 1 hypersensitivity reactions, everyone knows. Anaphylactic reactions. You don't need to worry about that. Anaphylactic reaction. Ethical clearance is not required in phase 1, phase 2, phase 3, phase 4. Clearance. Phase 1, yes. Which kind of ethical clearance? Phase 3, yes. Which kind of ethical clearance? Acha, you have to tell me both the ethical clearance. which kind of forms filling should be done at the time of initiating phase 1 and at the time of phase 3rd. Okay. Now, ethical clearance does not require in. So the answer is phase 4. You have not to require ethical clearance during the phase 4, during the post-marketing surveillance. Okay. Next and last question of today's class, that is majority of drug passes through biological membranes primarily by which transport? Passive diffusion, active transport, pinocytosis, facilitated diffusion. That is very easy for you. That is passive diffusion. Because 90% of the drug has a lipid solubility which can cross it through the membranes by passive diffusion. 90% of the drug passes through passive diffusion. Okay. This is also known as the downhill transportation. Okay. So this is all about the video. In this video, we have went through 20 important questions which are important for your GPAT typer of various pharmacy exams. So in the next video, we do have another general pharmacology part in which we will discuss another 20 questions from general pharmacology. Till then, keep revising and stay with GDC.